Good morning. All right, we're going to go ahead and get started right on time. So welcome to the session, Understanding HTLV Infection at the Molecular Level. My name is Amanda Panfil. I'm at The Ohio State University, and this is my co-chair, Dr. Lee Ratner from Washington University. So just a couple housekeeping announcements. Please put your phones on silent. As a reminder, audience, you can submit questions throughout the session using the Slido. Scan the QR code on the back of your mini program, or you can go to slido.com and enter hashtag HTLV. Selected questions will be put to the speakers after they have presented. And then what we're going to ask for each speaker, instead of sitting up front after you present, just take a seat in this front row. Then we'll all bring you up. That way you don't have to be on public display. All right. And for the first speaker, we have Estelle Plant from the Université Libre de Bruxelles in Belgium. They will be presenting identification of the cellular factor UHRF1 as a new epigenetic repressor of HTLV1 transcription.
So good morning, everyone. First, I would like to thank the organizer for letting me the opportunity to present my work on the role of the cellular factor UHRF1 in the transcriptional regulation of the HTLV1. So I don't have any conflict of interest. So as you know, the sense promoter is usually repressed in vivo, especially in ATL patients. This sense promoter is located in the 5' LTR. This led to the repression of the majority of the viral genes, except HBV. So there is no viremia, allowing the virus to escape the host immune system and to persist in vivo, and also to develop the HTLV1-associated disease. But it's not that simple, since there is still a strong, constant immune response against HTLV1, suggesting that there is an intermittent expression of the 5' LTR. Therefore, it is crucial to better understand the molecular mechanism involved in HTLV1 transcriptional regulation in order to develop new therapeutic strategies against HTLV1 and its associated disease. So the 5' LTR of HTLV1 is regulated by different factors, including transcription factor that can bind to cis-regulatory elements located in the 5' LTR. There is also the importance of the viral factor tags that can transactivate the promoter, but also the chromatin environment, including DNA methylation, histone methylation, and histone acetylation that plays a crucial role in this regulation. So in our laboratory, we focused our study on the cellular factor UHRF1, which is known to be involved in several cellular processes, including the cell cycle, DNA methylation maintenance, and DNA damage repair. But more importantly, UHRF1 is an epigenetic integrator that can link DNA methylation with histone post-translational post-translational modification through the interaction and recruitment of epigenetic enzymes such as the histone deacetylase HDAC1, the histone methyltransferase G9A, and the DNA methyltransferase DNMT1. So previously in our laboratory, we demonstrated the epigenetic repression of another retrovirus called the human immunodeficiency virus 1, HIV1, by UHRF1. Therefore, we wonder if UHRF1 could play this role also in the case of HTLV1. So first, we assess UHRF1 recruitment along the HTLV1 promoter, provirus, by performing chromatin immunoprecipitation experiment using the telohem1 cells that are an ATL-derived cell line where the 5' LTR is repressed. And we can see here that we have the recruitment of UHRF1 only at the 5' LTR. We performed the same experiment, but this time using PBMC isolated from ATL patients, and we also have the recruitment of UHRF1 only at the 5' LTR. Since UHRF1 is important for the recruitment of HDAC1, G9A, and DNMT1, we also assess the recruitment along the HTLV1 provirus. And similarly to UHRF1, we have the recruitment only at the 5' LTR, suggesting a repressive role of this factor in HTLV1 transcription. Concerning HDAC1, its recruitment and repressive role were previously demonstrated. We also assess the enrichment of the epigenetic marks associated with these epigenetic enzymes, and also the active epigenetic mark H3K4-methyl-3, which is usually present on active promoter. As you can see here, we have the enrichment of H3K4-methyl-3 to the 3' LTR, where we have the antisense promoter, which is always active. And we have an enrichment of histone acetylation at the 3' LTR, where HDAC1 was not present, and also the enrichment of the repressive epigenetic mark H3K9-methyl-2 at the 5' LTR, which this epigenetic mark is deposited by G9A. So we then assess the effect of UHRF1 on HTLV1 transcription by performing luciferase assay in HEK293 T cells. We use a reporter vector where we have the LTR 5' upstream of the firefly luciferase gene, and we co-transfect it with an increasing amount of plasmid expressing UHRF1. And as you can see here, when we overexpress UHRF1, we have a dose repression of the 5' LTR activity, suggesting a repressive role of UHRF1. We perform the same experiment, but this time we co-transfect the reporter vector with an expression vector of an shRNA targeting UHRF1. And when we depleted UHRF1, we have an increasing activity of the 5' LTR, demonstrating the repressive role of UHRF1 in transient transfection conditions. Now we want to assess the role of UHRF1 in a more physiologically relevant model. So we knocked down UHRF1 in two ATL-derived cell lines, the TELOM1 and ATL cells. And we looked at the expression of the viral transcript tags and GAG-propol by RT-qPCR. So we first validate UHRF1 knockdown by Western blots, but also by RT-qPCR. And when we have the knockdown of UHRF1, we can see a viral reactivation of the HTLV1 transcription, demonstrating the repression of UHRF1 on the HTLV1 5' LTR. Now the question is to know how UHRF1 repressed that promoter. Seeing its role as an epigenetic integrator, we hypothesized that UHRF1 could form an heterochromatin environment at the 5' LTR. So to test this hypothesis, we used the TELOM1 cells knockdown for UHRF1 and performed cheap qPCR experiment. As you can see here, when we lost the recruitment of UHRF1, we did not affect the recruitment of HDAC1, but we affect the recruitment of the DNMT1, and we actually lost the recruitment of G9A. We assessed in parallel their expression to see that the knockdown of UHRF1 did not affect the protein expression. And as you can see here, we do not have a change in their protein expression, even a slight increase for DNMT1. So the decrease that we observe here is really specific for their recruitment. Concerning the epigenetic mark, the H3K4-methyl-3 active epigenetic mark is increased following the knockdown of UHRF1 in accordance with the RT-qPCR results. We do not have any change for the acetylation, which is in accordance with HDAC1 recruitment that did not change, but we have a decrease for the H3K9-methyl-2 mark, repressive mark. Concerning the mark deposited by DNMT1, so DNA methylation, we perform sodium bisulfite conversion in the TELOM1 cells. So each line represents a clone, and each column represents a CPG position along the 5' LTR of HTLV1. And as previously reported, the HTLV1 5' LTR is hypermethylated in the TELOM1 cells. And when we induce the knockdown of UHRF1, we do not have a global DNA demethylation, but the specific demethylation of one CPG, the H CPG, which is present in a site that was identified as a net binding site. So UHRF1 could induce the specific DNA methylation and maybe impair the binding of important transcription factor for HTLV1 transcription. So finally, we also assess UHRF1 expression in individuals. We have uninfected individuals, people that are asymptomatic, and the people diagnosed with the neurodegenerative disease TSP or the cancer ATL. And what we observe is that UHRF1 is overexpressed in ATL patients, suggesting that maybe UHRF1 can be involved in ATL pathogenesis. We also wanted to see if there was a correlation between UHRF1 expression and the sense transcription of HTLV1. So it is represented here by tags. So we performed Spearman correlation analysis, and we showed that we have an inverse correlation between UHRF1 and tags expression that reinforce the results that we obtained in HTLV1 infected cells. So in conclusion, we demonstrate that UHRF1 is recruited to the HTLV1 5' LTR, that it can repress this promoter through epigenetic mechanism, through histone methylation and specific DNA methylation, and that UHRF1 could possibly be a potential new target for the development of new therapeutic strategies against HTLV1 in this associated disease. So I would like to thank my colleagues from my laboratory, but also our collaborators with Antoine Cheren, and also the French West Indies, and we have actually an ongoing project that will take place in September with them. And of course, the financial support, and also you for your attention. Thank you.
Thank you. The second speaker is Rochelle Zaldivar from Ohio State. We'll talk about SRPK1 interacting with HBZ messenger RNA and effects on cell proliferation.
Thank you so much for the introduction. Before I begin, I would like to state that I have no conflicts related to this presentation. And for my late summary, this project studies a viral molecule called HBZRNA. The HBZRNA is important for cell growth of HTLV1 infected cells. We have discovered that the HBZRNA interacts with a host protein in the cell called SRPK1, and we have found this interaction is important for promoting cell growth. As you know, HBZ is the only viral gene encoded by the antisense strand of the HTLV1 genome and is consistently expressed in all infected cells. Past studies by Pat Green's group demonstrated that shRNA-mediated knockdown of HBZ in both MT1 and newly immortalized PBL-ACH cell lines caused a significant decrease in cellular proliferation. This suggested that the HBZ played a role in promoting proliferation in infected T cells. Subsequent studies by Matsuoka further delineated the proliferative potential of HBZ lied within the HBZ RNA. Here they generate different HBZ constructs, HBZ, which produces both the protein and the RNA, a TTG mutant, which has a stop mutation at the start codon, and therefore only makes RNA, no protein, and an SM mutant, where the RNA was mutated at the third nucleotide and each amino acid, effectively disrupting RNA sequence, but not the protein. As you can see on the right, the TTG mutant, which expresses just the HBZ RNA, can induce proliferation, while the HBZ protein counteracts those effects. Based on these past studies, we hypothesize there are cellular protein interactions with the HBZ RNA reliant on RNA secondary structure, which translate to cell signaling pathways important for viral persistence and cellular proliferation. Shown here on the left is some RNA structure prediction using M-fold done in our lab, where we show the difference in predicted RNA secondary structure between wild type and SM HBZ mutant. As a reminder, the SM HBZ mutant will make protein, but has an altered RNA sequence. In order to identify the proteins which interact with the HBZ RNA, we took a proteomics-based approach. Using mass spectrometry, we actually got two hits. These were Angel2 and SRPK1. And Angel2 is a newly identified cellular protein, and the reagents available for our studies were limited. Therefore, today, I'm going to be focusing on SRPK1. We first validated our mass spec results using cross-linked immunoprecipitation, or CLIP experiments. We transfected 293 T cells with both an S-TOG SRPK1 expression vector and wild-type HBZ, or the SM-HBC construct. Then we pulled down using S-tag affinity beads and examined RNA, which is associated using RT-qPCR for HBZ mRNA. We found wild-type HBC, but not the SM-HBC interacted with SRPK1 as we expected. Then we used RT-qPCR and validated this in other cell lines that are physiological, the human T cell line KIT225s, and we did an IP for SRPK1 followed by RT-qPCR that identified an interaction with the HBC RNA. And finally, we confirmed this interaction in ATL patient-derived cell lines called ATLEDs and TLOM1s, and an IP for SRPK1 showed an interaction with HBC RNA. SRPK1 stands for serine arginine-rich splicing factor protein kinase 1. This is a cellular protein localized both in the nucleus and cytoplasm, where it regulates RNA splicing through phosphorylation of ASR protein splicing factors. It has also been shown to regulate the AKT signaling pathway and affect cellular proliferation. In order to test the effects of the HBZ mRNA SRPK1 interaction, we first needed to establish an HBC RNA proliferation assay. This was something that we really struggled with in the lab until a recent study from Matsuoka's group demonstrated that the HBC RNA is predominantly nuclear. So first, we generate several different constructs with varying lengths of poly A tails. One set has the standard SV40 poly A tail, while the other one has a short poly A tail. We also examined both wild-type HBC, which makes protein and RNA, and an HBZ TTG mutant, which will make RNA and not protein. Following on, we transfected these vectors into our human T-cell line KIT225s, where we find predominantly more nuclear HBC mRNA in the short poly A tail construct compared to the longer poly A tails from SV40. When we look at the proliferative effects of the short poly A tail constructs in the cells, we're able to observe that the HBC RNA proliferative effect is present with the short poly A tail. So using this system, we generate stable KIT225 cells, which express the HBC RNA only. We knocked down SRPK1 using shRNA and found the proliferative effect of the HBC RNA was lost. We also replicated these results in the ATL-derived T-cell line ATLED, and again showed that the loss of SRPK1 decreases proliferation in the cells. Based on what we know about HBC nuclear localization and proliferation, we next examined the cellular localization of HBZ RNA in KIT225 cells. Upon the SRPK1 knockdown, we see an increase in the cytoplasmic localization of HBC, which suggests that SRPK1 functions to regulate the cellular localization of HBC, thus regulating proliferation. In summary, we confirm that the HBC RNA mediates proliferation in T cells. We have identified a novel interaction between SRPK1 and HBC mRNA. Loss of SRPK1 ablates the proliferative effects mediated by HBC RNA, and this is likely due to the effect of SRPK1 on HBC cellular localization, shifting the abundance of HBC RNA from the nucleus to the cytoplasm. I would like to now thank all the members of the PanFL lab, and I'll also like to specifically acknowledge Dr. Patrick Green, as this is a close collaboration between our groups and is funded by our ongoing program project grant. Thank you so much.
Thank you. Our next speaker is Olivier Hermine from the University of Paris. He will be talking about targeting ATL with an anti-transferrin receptor.
So, good morning, everybody. So I will try to, and first I would like to thank the organizer to select or abstract for oral presentation. So it is a very nice collaborative work between several labs in Paris, here, in Pasteur Institute with a team of Antoine Gessin and Ambroise Marseille and Veida Snafi in NEM and my laboratory, and also a pharmaceutical company, biotech, which helped us to develop some antibody against the transferrin receptor. So, you know, we make a lot of progress in ATLL treatment, mainly due to stem cell transplantation. But one of the big problems we have today is to induce a complete response and disappearance of the clones before the transplantation. And in order to do so, we have developed a lot of chemotherapy, but I think most of these cells are resistant to chemotherapy, or subclones may emerge with resistance. And we try to overcome this resistance by using some antibodies, which is one of the strategies to clear ATLL cells. So we know maybe the transferrin receptor may be a good target for antibody. Why? Because it is a receptor which is able to uptake a transferrin from the peripheral blood, and transferrin bind iron. And upon internalization, the transferrin receptor release in lysosome compartment the iron, which serve for a lot of function in cells, which include metabolisms in the mitochondria, cell proliferation, and so on. And we know that tumoral cells, they need iron very heavily, and they do express highly the transferrin receptor to be able to uptake iron in the high content to face the metabolism change and the proliferation of the cells. And we have shown also that in addition, the transferrin receptor not only is able to bind transferrin and internalize transferrin with iron, but also with this small tail, we have shown that the transferrin receptor is able to induce a signaling pathway, because it has 4-serine and 1 tyrosine, which are able to bind some transition signaling pathway. And we have shown that it is able to induce the activation of the PI3 kinase AKT pathway and the MAP kinase pathway. So taken together, the transferrin receptor might be critical for the proliferation of tumoral cells, both to allow the proliferation and the metabolism change in the tumoral cells, but also to induce proliferation. So we have shown also that there is a clear correlation between the proliferation of tumoral cells and the expression of the transferrin receptor. And when we assess the proliferation by the Ki67 expression, you do see here a nice correlation between transferrin expression and Ki67 expression. So we have published a long time ago that, and also the Wallman group in NIH showed that the transferrin receptor is highly expressed on ATLL cells. And very interestingly, when we stimulate normal T cells with a high proliferation stimulus like PHA and IL-2, you see an increase of the CD25 and transferrin receptor, exactly what we do see in patients with cell line in ATLL. And interestingly, when you look at the chronic ATLL, which proliferate less than acute ATLL, you see the level of transferrin receptor is lower in chronic form of ATLL. So how to target the transferrin receptor? So we can envision, like you can see here, two possibilities is to bind and to prevent the binding of the transferrin to the receptor, which would be one of the possibilities. And the other possibility is to use some kind of ADCC or CDC approach to induce the stimulation of the immune system to get rid of the specific cells which express the transferrin receptor. But the second way is not very good, because you know transferrin receptor is expressed everywhere, not only in proliferating tumoral cells, but also in erythroblasts, in the bone marrow, very highly, but in the heart, in the GI tract, and so on. So to use ADCC is not a good idea with this targeting, because you may induce high inflammation in the heart and in the bone marrow. So you know that ADCC has been used and the strategy to block a ligand like IL-2 for ATL cell, but it doesn't work, as you know, because the the tumoral cells do not depend on IL-2 for their proliferation, and ADCC are not working, for example, with the CAMPAT, because CAMPAT induces a strong immune suppressive status in the patient, and we use also anti-CCR4, which may be better because it decreases the Tregs, which may increase also the immunity to increase the ADCC, but we know that the CCR4 might be downregulated in some instance, and they may escape also to the treatment. So, as you can see here, as a proof of concept, when you use an antibody preventing the binding of the TFR receptor, you see we induce a lack of the proliferation, and also we induce apoptosis of the cell through the mitochondrial depolarization, because we need, and we show that when you have induced massive deprivation of iron, you induce depolarization. So the next approach might be also to use ADC, and to bind some chemotherapy to antibody to target specifically the tumoral cells. And this one has two advantages, is that you bring the chemotherapy to the cells, and if the antibody is internalized, you will release the chemo within the cells and then release in the environment, which kills also other cells, even if they are negative for the target. So we try to see if we can use this approach, and brentuximab has been used, but also we know that patients relapse because they lose CD30, and CD30 is not a receptor which main goal is not to internalize, is to transduce signaling pathway, in contrast to transferrin receptor. So the caveat for using transferrin receptor is that we have to have an antibody which has higher affinities than the transferrin itself, and is able to induce the transferrin receptor and induce internalization if you want to use ADC, and it has to be safe in human because we know transferrin receptor is expressed everywhere. So first, what we did, we designed an antibody we call A24 from mice immunization, and we select one which was very interesting because a low affinity of transferrin receptor, it has a lower affinity than the transferrin in cells, meaning that the cells which express lower the transferrin receptor will not be targeted by this antibody. In contrast, in high expression of transferrin receptor, the antibody has a better affinity than the transferrin, as you can see in this biocore experiment. So we expect to see a selective targeting of cells which express highly transferrin receptor, but not the one with low expression. Second, we have shown that this antibody is able to be internalized in the endosomal compartment of the cells and remain at this level. So next, what we did, we humanized this antibody, and we use the IgG4 isotype to prevent, as I said before, ADCC, to prevent inflammatory reaction in normal cells. And you can see here the step of immunization of this antibody was very nice because we see the same capacity and the same properties of these antibodies. And then we add a chemotherapy, MMAE, and we use a new, we call APN linker, which is very, very selective and do not release MMAE in the peripheral blood, as you can see down when you compare to other antibodies with ADC. And then also this MMAE is bound to this linker, which is very sensitive to calpain, which is highly expressed in the compartment, endosomal compartment. So we tested this antibody in several cell lines, and you can see here, for example, in the leukemic cells, which express highly the transferrin receptor, that we do see an improvement of survival and response in this case. And interestingly here, we show that depending on the concentration and expression of the transferrin receptor, the cells are less sensitive to the antibody, and when we increase the transferrin concentration in the medium of culture, we do see that the antibody works less than when you have a low level of transferrin. And you can see here in the PDX model of triple negative breast cancer that depending on the level of expression of the transferrin receptor, the antibody works better when you have high expression than when you have a low expression of the transferrin receptor. And here we showed in another model of melanoma that we're able to block metastasis on this mice model when we use the antibody. And then what we develop cell lines TILL cell lines, which were resistant to brentuximab, because with a low dose of brentuximab, we select some cells which do express lower CD30 and become resistant to brentuximab. But in contrast, we have been never able to induce resistance to the transferrin receptor because the transferrin receptor remain expressed in the cells. So then we try to use, because I showed you before that we have a depolarization of the mitochondria, to see if we have a synergistic effect with BH3 inhibitors like venetoclax. And you can see here we have a nice synergistic effect between the antibody and venetoclax in several cell lines, including acute myelogenous leukemia. Then we move to the safety part of this antibody, because I told you transferrin receptor is expressed everywhere. And to do so, we did a double knock-in mice, in which we have changed the transferrin receptor, mice transferrin receptor to human, and transferrin mice to human transferrin. And you can see here, when we shift for transferrin mice receptor to human, you see a decrease of the PK of the antibody, because it binds to the human but not to the mice transferrin receptor. We didn't see any safety issue in this double knock-in mice. And then we move to monkeys, and then we look very carefully at the heart, because transferrin receptor is expressed, and no safety issue in mice. So to see T cells, because I told you they get express highly the transferrin receptor. But interestingly, when we use CD30, CD3, CD28 stimulation, we see an increased expression of transferrin receptor, but to a lower level, which is not targeted by the antibody when you compare to the transferrin, meaning that we have no toxicity to T cells even when they get activated. Obviously, when we do strong activation with PHA-IL2, we see high expression of transferrin receptor and high toxicity of the antibody. So then we move to the phase 1 study in patients with acute leukemia. And as you can see here, we do see some response, and we have defined the MTD in this patient. And you can see here, one of the patients will enter in complete response and improve his hematopoiesis. And to finish, I'm sorry, I'm late. We develop a PDX model, which will be presented this afternoon by Ambroise. And in this model, we could prevent the development of ATLL, and it's a very aggressive model of ATLL. And when we do see relapse after one injection, we look at the transferrin receptor expression, and it's still very high in this cell. So we retreat, and we see again a downregulation of the proliferation of the cells. So taken together, we strongly believe that transferrin receptor with this antibody, with high affinity to the transferrin receptor in competition with the transferrin, might be used in ATLL to induce a better response rate in combination, for example, with venetoclax. So I'm sorry to have been late, and thank you very much for your attention.
Thank you. Next speaker is Pooja Jain, who will talk about long-distance control of immunity in ham TSP via exosomes.
Okay. Hello, everyone, and thank you, organizer. I don't want to hear that alarm, so I'm trying to rush a little bit, so I'm sorry. I'm also apologizing for some busy slides, because I have to tell you a story, so it's going to be, so please forgive me if you feel like some of the slides were too crowded. Anyway, so what I'm going to share with you is a continuation of my talk in workshop, so but today's database, and provide evidence and convince you how the extracellular vesicles can be a way for the HTLV infected cells to attract more cells in their parameter, and also to affect the immunity, which has been a, the issue in our field, especially with respect to ham TSP for a long time. So obviously I have no conflict of interest, and I feel sorry about that too. Anyway, so some labs, including our labs, have been trying to delve into complexity of this CD8 T cell response during ham TSP, because we know the goal of vaccine is to have a large number of antigen-specific T cells, which we have in ham patient, but still the patient keep very high proviral load and develop to disease. So why? So from very beginning, we have done some very extensive in vivo work. So we created CD11C DTR transgenic mice and cross-bred them with HLA-A2. So we can knock out dendritic cell and understand the priming of response since inception. And taking lead from our in vivo studies which have been published, we went to about a cohort of 100 patients from Jamaica and further solidify our observation from in vivo models, as well as we have extensive cell culture system with primary cells of dendritic cells and T cell to understand what's going on. So combining all those prior work, we showed that both the tax protein and the virus itself is capable of mounting very strong response. In fact, through immature dendritic cell, which is the most potent antigen presenting cell, these cells do undergo the process of activation and maturation and lead to the oligoclonal expansion of CD8 T cells by the presentation of tax peptide. So these studies were heavily focused on tax immunodominant peptide in HLA-A2 patients and in the HLA-A2 system, but now we have expanded to much bigger repertoire of epitopes. So what we propose and what we think that in case of the people who are able to control the infection, this immune response is very effective because it's able to clear the antigen, and due to lack of antigen, the phenotype that we get in CD8 T cell is an effector phenotype. However, in the chronic patient, especially those develop HAM-TSP, because of the consistent presence of low-dose antigen, this effector response becomes dysfunctional or exhausted over the period of time. So we know from the well-established field of LCMV and cancer, and now with our work and other work, that these exhaustion phenomena is very apparent in terms of chronic viral infection, which is mounted by immune checkpoint molecule, which are essential to bring the T cells to activation status. However, if the antigen remain persistent, both the immune checkpoint receptor and ligand remain expressed on the surface of these exhausted T cells, and some of these do constitute central memory. So the interplay of these immune checkpoint ligands on antigen presenting cells and their receptors on T cell is essentially the one that dictating how the immunity will unfold, especially with respect to adaptive immune response. So here I'm just showing you data from our recent Brazilian cohort where we show that indeed upon the recall responses, the antigen-specific T cell in HAM-TSP patient do express higher load of several immune checkpoint receptors, and it is true for CD4 as well as CD8 T cells, but they also express dual or triple or more immune exhaustion molecule. In fact, there is a direct correlation between the expression of PD-1, which is one of the well-known immune checkpoint molecule, and the efficacy of CD8 T cell response, which negatively correlate with the PD-1 expression. So there is from the cohort studies, we can say for sure that there is a direct effect of these immune checkpoint molecule. Now, the latest data which is coming in the field suggests that these ICPs don't have to work just to be mounted on the cell surface, because they can exist in a soluble cell-free form and in fact can be carried on the surface of extracellular vesicles and exosome, which you can call like small EVs. So the difference between the membranous ICP and soluble ICP is not that much, because in terms of structure they are very similar. The only difference is that these ICPs are actively spliced during the process of transcription and translation, and then they lose their N-terminal signal. So the only ways for the soluble immune checkpoint to be transported on the membrane is through become a cargo on the vesicles that are constantly being released by the cells. Okay. So how these exosomes are made, like I say, it's a natural process of cellular secretory machinery where early endosomes in cooperation with late endosome merge into multivesicular bodies, and these multivesicular bodies, when they fuse and egress from the cell surface, they express a plethora of molecules including adhesions and their own escort protein, CD81, CD9, CD16, as well as immune checkpoint molecules and mRNA, microRNA, and cytokines, and so on, as well the viral protein. So what we hypothesize that HTLV infection-derived exosomes can carry immune checkpoint molecules that maintain their immune modulatory activity, and they can serve as diagnostic marker as well as therapeutic target to combat the immune exhaustion seen in HAM-TSP. So we started this work from using the samples we got from Ed Murphy's lab, and we showed that compared to seronegative control or HTLV positive asymptomatic carrier, the HAM-TSP patients were expressing in their serum a number of soluble ICPs, and some of the ICPs that we found were very unique and interesting, such as PDL2. So we didn't notice any significant effect, surprisingly, in the soluble form of PD1, but we noticed that PDL2 was extensively expressed on these HAM-TSP cells, so as a number of other soluble ICPs. So the profile was very clear. So then we proceeded to produce the exosome from the sera of HAM-TSP patient. We used either the ultracentrifugation method or the ISON column, and like I was telling you, the ISON column was fantastic in terms of producing large number of exosomes from the patient sera. So this, after producing the exosomes from both HTLV1 and HTLV2 infected patients and asymptomatic carrier, we just characterize them by a number of methods. NanoTrack is a small video of showing, but it's okay. We can pass on just showing the well-formed extracellular vesicle and their movement by electron microscopy, and also by this new technology called ExoView or NanoView, in which you can look at individual exosomes in correlation with the expression of the immune checkpoint molecules. And obviously, we confirm the presence of the escort protein on the isolated exosomes and the presence of selected ICP molecule that we screen for from the sera. So I'm bypassing a lot of data in between and just going to the observation that we feel is very striking. So we asked the question, what is the consequence of these exosomes from the perspective of immune responses? So we started exposing exosomes to the patient-derived CD4, CD8 T cells as well as dendritic cells. So we have two different models, either to expose directly or through dendritic cell, which obviously our base cell that we have been working for 20 years now. So the reason for dendritic cell, because we know that they can polarize naive T cells to a very different phenotypes, and as you know, for HTLV1 infection, the sum of these Th1 Treg population could be extremely important. So here, the first effect of exosomes on myeloid dendritic cell and plasmacytoid dendritic cells, we notice definitely exosomes are able to activate DC in terms of stimulating CD40, 8086 molecule. And by MSD analysis, the mesoscale discovery, we identified the unique cytokine on MDC, PDC. They come out upon exosome exposure. These include TGF-beta, IL-13, IL-6, and TNF-alpha. And then we use a co-culture technology where use the exosomes and pulse dendritic cells and then mix with the syngeneic T cells and monitor the Th1, Th2, Th17 and Treg phenotype. So here you can clearly see that upon exosome exposure, we have a significant increase in interferon gamma CCR5 expressing Th1 and CD25 FOXP3 expressing Treg, and which was also true in terms of the release of those specific cytokines that skew this phenotype. Thereafter, we basically see if this process is also governed by the immune checkpoint. So here we are doing two different experiments in the healthy cells and as well as in the patient-derived T cells. So in healthy cell, we have to activate our T cells. We did by many different ways. The best effect came from PHA-activated T cells. So you are seeing that OSP2 derived exosomes do down-regulate the CD8 T cell functional marker, and that can be restored by ICP blockade. And very similar effect we notice with the HAM-TSP derived T cells, that exosomes do suppress their immunity, and we can restore that by the immune checkpoint blockade. So again, because of the time, I'm not showing you the mechanism, but this is actually done by exosomes to affect the signaling of immune checkpoint via PAKT and also by HBZ. So HBZ story is published. You are welcome to look at it because I don't have time to share with you. But a very quick note about the mechanism. So this is a very complicated model. We're still trying to solve it, but it happens actually the interaction of TRKB with HBZ on ISRE promoter that interfaces with NF-kappa B signaling, affects ICP transcription and cytokine secretion, and that could be the reason for higher level of ICP we see in HAM-TSP. So this is my layman summary and conclusion. I guess I'm gonna pass it. I don't want to keep Amanda and Lee like, because I have to thank my large list of collaborators. And so this was pretty much the work of a PhD student, Julie Joseph, who has moved on. So we have a position for postdoctoral fellow. If anybody interested, contact me. And I want to thank my collaborator, Lish and Fatah, who contributed to this work, and Ed Murphy for the patient sample. Thank you for your attention.
Well, if the speakers could join us in the front of the room, we have numerous questions. So I can start out with the first speaker, Dr. Plant. So questions about UHRF1, whether its activity is dependent upon the ubiquitin ligase activity, and what would be the target for ubiquitination?
Can you repeat the first part of the question, please?
Question, so you showed it's a ubiquitin ligase. So is the ligase activity required for its activity on HTLV1 infected cells? And what is the protein target that is ubiquitinated, if that is true?
For now, for the basal activity of for the activity of UHRF1 on the basal transcription of HTLV1, the ubiquitination activity of UHRF1 do not take part. It's just the recruitment of epigenetic enzymes, but we have preliminary results for the transactivation conditions, where UHRF1 might ubiquitinate tags.
So we have a question from the online. So to Ms. Zaldivar, what is the expression of SRPK1 in primary infected or ATL cells? And then a second part, does knockdown in primary cells reduce their proliferation capacity?
We have looked at the expression of SRPK1 through Western blotting in newly infected cells and also in different transformed and ATL-derived cell lines. And what we found is that there is no difference in SRPK1 expression. And for the second part, we have actually knocked down SRPK1 in KIT225s and JERKIT parental cell lines. And we have not seen that decrease in cell proliferation. This is an event that we only see when we knock down SRPK1 in the presence of HBC RNA.
So for Dr. Hermine, there are actually quite a few clinical trials already underway with anti-transferrin receptor antibodies. So are there any preliminary data, and why did you have to develop a new monoclonal antibody since there are many other commercial products that are already being used in clinical trials?
To my knowledge, there is no approved antibodies against transferrin receptor. There's only one, which is the new technology, which was cytomix, in which they protect the transferrin binding site, which get activated only in the vicinity of the tumoral cells because there is some kind of cleavage proteins. But the problem is, in this setting, that some tumoral cells do not express the protease to activate the antibody. But then when it becomes activated, because it's not in competition with the transferrin receptor, it binds all tissues and lead to some toxicities in ADC settings. So they show that in the phase 1 trials in lung cancer, they show some response, but because of toxicity and particularly anemia, they stop the development of these antibodies. So currently there is no antibody except the one that we are trying to develop now.
I think we'll take one from the audience.
Yeah, I had actually a question for Estelle. It was a great talk. But I had a question about the specificity of that one specific CpG site that you saw demethylated or a reduction of methylation in your knockdown. Given what we know about the mechanism of action of UHRF1 with maintenance of DNA methylation, it's quite unexpected. So do you have any thoughts on the specificity as to maybe how or why it's that one site?
Thank you. It's a good question, actually. We are still wondering how UHRF1 can be that specific. We don't know if it's, we just tried to knock down UHRF1 and we saw that one CpG was demethylated. But the telomere-M1 cells are really repressed, strongly repressed. So it's really hard to reactivate them. But what we could try in the future is to actually overexpress UHF1 in productive cell lines where they are not methylated and see if they can induce more methylation. But for now, we only have that CPG, but even with just the demethylation of that one specific CPG, we already have a reactivation. So that shows that UHF1 is really important for the regulation of HTLV1.
All right. Go ahead, Pat.
Yes, I have a question for Dr. Simone. So maybe you mentioned this, but if you prevent the P12 cleavage by mutation in the virus, what's the phenotype of the virus in vitro and in vivo? Do you know that?
So here's the problem. I'm working with the HA-tagged construct to detect it in vitro. Obviously in vivo it's not HA-tagged, and there's no specific antibody for it. So when I use it in my in vitro experiments, I usually get 60% P12 and 40% of cleavage to P8 when I transfect it in 293 T cells. And it's comparable in Joker T cells as well.
Let me follow that up with a question from the audience asking whether you specifically quantitated the number of conduits in the cell-to-cell contacts in that transmission assay that you showed?
So the number of conduits I showed was from a publication from the Franchini group. So I haven't done it yet, but that's ongoing. So I mean, ultimately, that's the underlying mechanism that I propose, that the interference with the cell-to-cell transmission is dependent on the cellular conduit. So I obviously have to show that it's really a decrease in the conduit formation, and that's ongoing.
Charles?
Hi, thanks. Question for the last speaker. Your cluster analysis showed that circular RNAs looked as though they were associated with donors and separated them from ATL patients. But your general RNA-seq analysis also showed that they were separated from in the same group. So how can you be sure that the circular RNAs are genuinely specific to ATL rather than just infection?
Relevant to the subtype you are talking about.
Sorry?
Relevant to the differency of the subtype you're talking.
Okay, but they may be that may just reflect the RNA expression in the subtype.
The RNA. Excuse me. Can you repeat the question?
I think we better pursue this over coffee.
Yeah.
It's more complicated than that.
Repetition, if you.
Yeah. It's okay. We'll pursue it over coffee.
Okay, we'll go back to some online here. So for Dr. Jane, what method did you use to identify HTLV1 specific CD4 and CD8 T cells in the Brazilian cohort?
So there were direct and indirect. So direct method, we have created tetramers. In fact, we are in the process of generating tetramer for about 30 neoepitopes that we have identified, and so that's directly we can identify through tetramer, but the indirect method we relied on the recall responses. So recall responses, we have created 80 overlapping epitopes from the entire text sequences because we didn't want to miss out anything, and then went in exposing our PBMC with those the pool of 80 overlapping peptides to generate the recall responses. So depend upon the number of cells and depend upon the status of the cryopreserved PBMCs, we have to rely on either direct or indirect or sometimes both methods to confirm tax specificity.
Okay, all right.
Oops. Sorry, so I have one comment and two questions, if it is possible. So one comment is in response to the question that was made before about the defect in the cleavage of P8. We have shown that in vivo, if you do that, you don't infect macaques, number one. And number two, if you now look at the infected individual and you ask the question, if there are mutations in the cleavage site, can you actually see a difference in the virus load? And the answer is yes. If you have a mutation in the P8, in the cleavage site that makes the P8 less cleaved, you have actually a decreased virus load. So I think this is some of the suggestion. But I have a couple of questions. So one question is related to the SRPK1, And my question for you is, if you do take now infected cells and you silence the expression of this, can you see a difference in splicing, in the splicing of different messenger RNA?
We have not looked at the splicing yet, but this is something that we definitely are looking forward to. It is important since, like I mentioned during the presentation, that SRPK1 has been shown to be very important in splicing. We are definitely interested in looking at that, as well as the kinase activity of SRPK1. So I don't have the answer right now, but for sure, hopefully in the future, we will.
And the second question is for the next to be Dr. King, which is, if you, do you look to see if when you silence YTHDC1, if you do that, do you have an effect of P30 on this? Did you look at P30? You know, as you know, P30 is affecting the transfer of the taxarex messenger RNA specifically. So I was wondering whether if you now put everything in the context of an infection, what would you see?
That's a great question. Thank you for that. I have not looked at P30 yet. Are you just thinking about P30 in terms of transcript abundance or localization? Yeah, I have not looked at either yet. Similar to Ms. Zaldivar, that is a future plan of mine.
A speaker or a question from a current
Yes,
speaker?
I have a question about your presentation of VEGF and KDR interactions. And you show nicely when you use, like, I think it was a SUTENT kinase inhibitors, you induce autophagy and degradation of tags and then apoptosis of cell lines. Did you try to use antibody against VEGF to block the interaction between KDR and VEGF? Because Ali have shown long time ago that there's an autocrine loop. And do you see a differential effect between you using an antibody or using kinase inhibition? Because SUTENT is quite a dirty kinase inhibitor. It blocks VEGF, but like 20 kinases more. So did you see it?
Right, so you're asking if I used VEGF, adding
VEGF
VEGF
antibody
to cells?
to block the interactions between VEGF and KDR, or if you use only the kinase inhibition, because you may have an effect because you block indeed KDR, but also additional kinases. But if you block the interaction between VEGF and KDR, it's more specific of what you do see.
Yeah, we haven't tried that experiment because from our imaging experiments, it seems like KDR is inside the cell in the area of the Golgi. And so we think if we add ligand that it won't have any effect since it's interacting with TAX at the Golgi. But that's something that we should probably try to see if VEGF has any effect on that. So
But KDR is phosphorylated inside the cells?
Yeah, KDR is inside, so it normally traffics from the plasma membrane to the Golgi. Okay. Usually at the Golgi, it's inactive, it's simulated. But in this case, we think TAX is keeping it active at the Golgi.
Okay. And actually, I have a question related to that. So I didn't know the specificity of this SU1498. I mean, cabozantinib is a very nonspecific kinase inhibitor. There is an FDA-approved antibody directed against the VEGF-R2, ramucirumab, and I wonder if you've also tested that.
Yeah, that's a good question, Lee. We haven't tested the antibody. Again, we think KDR is inside the cell, so I don't know how well the antibody would work. I think the small molecule inhibitors would have a better effect than a monoclonal antibody, but I mean, it's something we can try. But we have tried 3 or 4 different KDR inhibitors, including the FDA-approved one, cabozantinib, and they all have the same effect. They all induce TAX degradation.
And specificity of the drug that you used most on your study?
So the one we mainly used was SU1498, and that's the most specific one for KDR. Cabozantinib, as you mentioned, can inhibit MET and other tyrosine kinases.
Okay. I have a question for Dr. Takatori. So you actually looked at lymph nodes and PBMCs from 3 acute patients, but it would be particularly interesting to look at lymphoma subtype and whether the genes upregulated in the lymph node in the acute patients, how does that compare to the genes that are upregulated in lymphoma tissue from a lymphoma subtype of ATL?
Other subtypes ATL. I haven't checked yet, but I'm trying, because actually I'm in the middle of analyzing. So, but in the near future, I will check.
Take a speaker or a question from the audience.
Amazing selection of talks, and it's actually another question for Dr. Takatori. I was just wondering, with your methods, would you be able to look at the kind of different expansion of cell populations in specific body compartments if we think about transmission?
Sorry, expand it.
So obviously you've focused on ATL and the kind of expansion of ATL cells, kind of comparing PBMCs and lymph. Would you be able to do it with kind of non-ATL infected cells, thinking about kind of transmission with specific body compartments? So kind of expansion within, say, semen or breast milk or cervicovaginal fluids?
Sorry, I don't know what you mean.
I really enjoyed with your work the way you looked at the kind of progression of the kind of cell changes with the PBMCs compared to lymph. Could you do it with non-ATL, just infected cells, to see if there's kind of specific expansion in particular body compartments when we think about transmission from those compartments?
In this study, we roughly check the pseudotime course. Then, so I don't know how to say. It's extremely final upregulated gene in tumor cells, in lymph node or peripheral blood. But this is between the difference between tumor peripheral blood and lymph node. The final, extremely the last, at the end of the pseudotime phase. Is it correct?
We can chat.
Yeah, maybe we'll continue that one after. We're going to take one last question. I know we're a tiny bit over, but one last question. And I know there's a lot also on the app that you guys can take a look at too.
And we'd recommend that the speakers stay in the front because we have several more questions that we don't have time to ask, but maybe people can come up and ask them specifically. So. Last question.
Thank you very much. Question for Dr. Plant, please. Do you have any thoughts about the specificity of binding to 5' LTR as opposed to 3' LTR for UHRF?
Can you repeat, please?
Yeah. Do you have any thoughts about why you saw specificity of binding of UHRF to 5' as opposed to 3' LTR?
But the first thought was that the 5' LTR is repressed, and one mechanism of UHF1 to bind the DNA is actually if there is already some methylation. He has an SRA domain that is really important for its binding to DNA. So I suppose that since there is no DNA methylation at the 3' LTR, there is no way that UHF1 can be recruited to it. So that's why I think it's mainly at the 5' LTR.
All right. Thank you. Thank you to all of our wonderful speakers. Thank you to the audience.